Assalamu alaikum. Dear students, this is me, Atiq Ahmad. Today we are going to study the topic cytokinesis. In the previous lecture, we have studied the topic karyokinesis. We have studied the four phases of karyokinesis. Today we will study the cytokinesis, the division of the cytoplasm. In both animals and plants, the process of cytokinesis is different. Cytokinesis. First we will study cytokinesis in animals. Then we will study cytokinesis in plants. So in animals, after the division of the nucleus, the cytoplasm divides. After the division of the nucleus, we have already studied the four phases of the karyokinesis in which the nuclei is divided. So after that division, the cytoplasm will be divided. The division of cytoplasm is called cytokinesis. In animal cells, cleavage or furrows appears in the cytoplasm between the two nuclei. This is one nuclei, this is another nuclei. In between these two nuclei, a furrow, a cleavage will appear. Now it is shown here in this diagram where clearly this is animal cell. So these are the two nuclei which are already formed in the karyokinesis. In the cytokinesis, cleavage furrow, this cleavage means breakage. This cleavage furrow, this cleavage deepening will appear from this side and from this side. This deepening, this furrow will appear and it will then deepen down and the two cells will be formed. One cell will be divided into two. So in animal cells, cleavage or furrows appear in the cytoplasm between the two nuclei. Between these two nuclei, a furrow will appear. We have already seen that in the other diagram. The furrow will deepen and the two identical daughter cells are finally produced. And these are the two identical cells which are produced. And we have seen in this diagram, these are the two nuclei. The cleavage furrow will appear and it will deepen down and then it will divide this one cell into these two cells. In case of plants, in plant cells, a cell plate is formed between the two daughter nuclei. This is one nuclei, this is another nuclei. In between these two, the cell plate, this cell plate is formed. This cell plate is basically formed from the vesicles. The vesicles are produced by the Golgi apparatus. It divides the cell into two. The cell plate is formed by the fusion of small fluid-filled vesicle produced by the Golgi apparatus. So in case of plants, first the karyokinesis will occur. So these two nuclei are formed. This is one nuclei, this is another nuclei. Then in between there will be vesicles. These vesicles are produced by the Golgi apparatus. So these vesicles will fuse with each other. So this, in the second stage, a cell plate is formed. This cell plate will divide the one cell into these two cells. This is one cell and this is another cell. This is also shown here in this diagram. This is plant cell. This is one nuclei. This is another nuclei. Nuclei is already divided. In between, these are Golgi vesicles. These are vesicles. These are small pouches. They will combine together. They will fuse with each other to form a plate. This cell plate is formed and this cell plate will divide one cell into these two cells. So this is all about today's lecture. See you next time with new lecture. Take care. Allah Hafiz.